Welcome, dear participants, to the national webinar on the role of transgenic fishes in drug discovery and the scope for entrepreneurship. Now, I request Dr. LRS Kalanidhi, Vice Principal, Students Affairs, Guru Nanak College, to kindly give the welcome address. Ma'am, please.
Good day to one and all present for today's national webinar on the role of transgenic fishes in drug industry and the scope for entrepreneurship, organized today, the 1st of October 2020 by Guru Nanak Center for Research, GNCR, Guru Nanak College, Velachery, Chennai. I deem it a privilege to welcome this august gathering. On behalf of our management, principal, staff, and Guru Nanak Center for Research, I heartily welcome our most distinguished resource person, the Professor and Head, Center for Marine Science and Technology, Manonmaniam Sundarnar University, Tirunelveli, Dr. S.G. Prakash Vincent, who has many feathers to his cap. He is a DAAD Fellow Germany, EMBOWP Fellow Germany, UNESCO Fellowship Awardee Italy, Marie Curie Fellow EMBL, Chairman Associate BRICS Biomed Consortium, and Focal Point European Commission Horizon 2020. Hearty welcome to you, sir. Our Guru Nanak College, along with Guru Nanak Center for Research, feels honored to have you in our midst. Hearty welcome to you, sir. I am delighted to welcome our Honorable General Secretary and Correspondent, Mr. Manjit Singh Nair, the Chief Patron of all our academic endeavors. Hearty welcome to you, sir. It's my pleasure to welcome our most respected principal, Dr. N.G. Raghunathan, who steers us with his foresight and sharp caliber. Hearty welcome to you, sir. I welcome the convener of this one-day national webinar, the Head Department of Advanced Zoology and Biotechnology and Dean Research, Dr. J. Jayanti. During lockdown period and till date, she has been organizing national and international webinars, FDPs, conclaves, workshops, and so on in all disciplines, enhancing the quality of research and entrepreneurship. She keeps us all engaged throughout by opening new vistas. Hearty welcome to you, madam. I welcome the organizing secretary of this national webinar, Dr. Mahendra Kumar, Assistant Professor of Biotechnology. He has been regularly contributing his voluntary support in all our programs, not only for GNCR and his department, but also for all of us in other departments. Hearty welcome to you, sir. It was nice discussing with you yesterday when I asked you a question out of curiosity about the topic of today. And you very interestingly, you shared with me that from 2003, more than 35 species of transgenic fish have been genetically engineered in research labs worldwide. And the names like carp, tilapia, salmon, channel catfish, white zebrafish, medaca, and goldfish, they all sound very interesting. Thank you, Dr. Mahendra Kumar, for giving me that small orientation. And hearty welcome to you. I welcome our controller of examinations, all the deans of 10 schools, heads of all departments and members of other departments, research scholars and PG students of Guru Nanak College. On behalf of GNCR, Guru Nanak Center for Research and our college, I cordially welcome the learned professors, scientists, and all members across the world who have registered in our Telegram link of our Guru Nanak Center for Research. Hearty welcome to all ladies and gentlemen. I wish the program a grand success and thank you for the opportunity given to me. Hearty welcome to you all once again.
Thank you very much, ma'am. I deem it a pleasure and a proud privilege to introduce our chief guest today. Since sir needs no introduction, it's a formal gesture to introduce the chief guest. Dr. S.G. Prakash Vincent is the head Center for Marine Science and Technology, Manonmaniam Sundarnar University, Kanyakumari, Rajakamangalam Campus, Tamil Nadu, India. Dr. S.G. Prakash Vincent is designated as Chairman Associate of BRICS Biomed Corporation in 2014. He is also serving as focal point of European Commission for its Horizon 2020 for the period of 2014 to 2020, research funding to foster Indo-European Union collaborations. Horizon 2020 has a funding budget of 70 billion euros, and he is the coordinator of the International Center for Biotechnology, ICN, Manonmaniam Sundarnar University. He is a DAD Fellow, EMBO Fellow World Program, Marie Curie Fellow European Commission, and UNESCO Biotechnology Fellowship awardee. He had his research and training undergone in 4 universities and 2 research centers in Europe in a total period of 5 years, since 1994. He was part of the Indian scientific delegation to visit Sofia Antipolis Park, Nice, France in 2004. He undertook his Marie Curie IIF project at European Molecular Biology Organization, Heidelberg, Germany from 2004 to 2006. His area of work is drug discovery. He has guided 6 PhD scholars. He has 35 publications, 4 books, and delivered about 40 invited lectures in nanobiotechnology, functional genomics, transgenics, etc. He had organized European Molecular Biology Organization sponsored World Practical Course on DNA microarray analysis and applications 2 times in India. Through this program, the International Center for Nanobiotechnology has alumni of 17 nationals. He has a project funding of more than 2 crores to his credit. He has organized several international and national training programs through which many life sciences students, postgraduate students, research scholars, and faculties has got benefited. He has got a big list of publications, and Sir is one of the very few nanobiotechnologists, celebrated nanobiotechnologists of our country. He has traveled to more than 13 countries on official assignments. Sir, we feel very proud and happy to have you as a chief resource person for this national webinar on role of transgenic fishes in drug discovery and the scope for entrepreneurship. Now over to you, sir.
Thank you, Dr. Mahendran. Good morning to all of the participants. Here it's my pleasure to present in front of you. I'd like to thank the secretary, the principal, vice principal, the deans, and the current dean, and Dr. Mahendran for giving me the opportunity. I'm presenting from Kanyakumari district, and I hope that there are a lot many participants. I'm delighted, just thrilled to hear that so many 800 registrations are there. And I hope that this talk will be useful to you, and many will take leads and at least start thinking on different lights of drug discovery using transgenic fish. And can I go with the first slide? Is it the first slide?
Sir, you can open your PPT, sir.
Okay, 1 minute.
Yes, sir. Yes, sir.
Wait 1 minute. I just put my first slide. Has this come full screen now?
Yes, sir. Yes, sir. It's full screen, sir.
Okay. Anyway, good morning to all. So this is the title of my talk, like role of transgenic fish in drug discovery and the scope for entrepreneurship. Okay. And as a content, this is a little technical. You need to bear with me, and because in the final part I'll tell about the entrepreneurship opportunity. So I'll be talking on drug discovery overview and zebrafish or Medicus related to zebrafish as vertebrate models, advantages of having zebrafish as a model. Then I'll talk what is a gene knock-in and how I did a gene knock-in at EMBL Heidelberg, the construction of transgenic vectors, microinjection. I'll touch upon in vivo live confocal imaging, how then a technique of in situ hybridization, antisense RNA-based knockdown, then stable gene knockouts in zebrafish. And then next part will be transgenic fish models for human cancer, organ toxicity assays, angiogenesis, and high throughput screen, how it's done by zebrafish in the drug discovery platform. Coming to entrepreneurial prospects, I'll be in between also I'll be telling about identifying a novel human disease targets using zebrafish, identification of human therapeutic drugs, novel peptides. Then what is the prospects of mass production of biologicals using zebrafish, etc., will be covered in my talk. Just coming to overview of drug discovery, this is the total list of things which is done in drug discovery. Normally it takes 15 years. If you see that one drug to come to market, a company spends like $2.8 billion US dollar or 18,000 crores to bring a drug to market. And now, two aspects are here when you tell about drug discovery. One is called a target, okay, and another is called hit or lead molecules. These two are the important aspects when coming to the targets of protein molecules present in our body. It can be in the heart, it can be the liver, okay, it can be anywhere, kidney or in the muscle. It's a different protein molecule because we have like 20 to 25,000 genes expressed in our body. Each gene is expressed as a protein molecule, so it can go. So target is a protein molecule which is found in the body. Hit or lead is a small molecules which can bind to targets, and this is from it can be a microbial source or a plant source or some even from an insect source. A hit molecule, small molecules. If you see the drugs which we are taking, if you go to a medical shop and see the tablets, the 50% hit molecules, small molecules, the drugs come from actinomyces, a bacteria, soil bacteria. It's like amazing, 50% of the drugs comes from actinomyces. So there's not scope to screen for small molecules in from actinomyces. So this is what we are doing. The red arrows shows the areas which we are doing in our lab, trying to identify novel molecules, and we have come out with a novel molecule called alageninthysin from a marine bacteria actinomyces, which is active against human superbugs, particularly MRSA. We published in Journal of Antibiotics, and we use zebrafish for some screening of the active molecules. Okay, so now this comes like a target discovery. Comes like overview. I'm not going to explain totally on this thing, but I just give you an overview. You see, target types and druggability, function, genomics, knock-in, knockouts, gain of function, pathways, clinical data, all comes up target discovery, target validation, assay development using different models. Rat is model, we use even insect Drosophila use, and zebrafish or Medica is emerging as a good model at this time. Then you can have screen hits to lead molecules. We can have for synthesis of molecules or take crude extract. You have seen many of the experiments taking crude extract and trying to assay it. Okay. Then once from a lead, then if the 1,000 leads are there, few will come. 1,000 hits are there, and few molecules will come as lead molecules, proficiency molecules. Then they will go for preclinical study, lead optimization. Once development, then it goes for FDA approval. Then it goes for clinical studies, phase 1, phase 2, phase 3. And the COVID virus, some antibodies or small molecules are already in phase 2 and phase 3. So this is the total aspect of the drug discovery. Here we are going to see wherever this zebrafish is used in the drug discovery process, and particularly a transgenic zebrafish. I will tell you why transgenic we need and what is the advantage of this zebrafish in my later part of my talk. So now, if you see that this is the human body, okay, this is the zebrafish embryo. Now you can see the eye, the letter. So see that zebrafish have 72% of orthologous gene in humans. So if 100 genes are involved in the central nervous system development for human, 72 genes are already there in the zebrafish. Okay, in totally just you can take even all the organ development, you can see that 72%. So if some molecules can enhance the eye vision in a zebrafish, automatically that molecules also will work in human. Same with heart. Okay, if there are some molecules which can increase the pumping rate of heart, then that is a good molecule for a promising molecule for use in humans. So this is why we use a zebrafish for large scale screening. Okay, then so this slides, I think the slides will also be shared to you. I'm not going to the much details. Here is the fly, zebrafish, mouse, rat. What are the advantages? No? Plus is the lot of advantage. If you see this transient in vivo screening, like zebrafish is one of the top, no? Okay, because of the 4D imaging, no? You can put a zebrafish under a microscope and then go for imaging. You can see all the organ development. Okay, once you transgenic label, individually you can label the brain, you can label the heart, you can label the pancreas. Okay, and then see it live under the microscope. So the development process. So you can make a knockout of a disease model and then screen and then find out molecules which can rescue the knockouts. So this way, zebrafish is used in a very robust manner all over the world for in the drug discovery process. Now, one more point, here, this zebrafish. Okay, this size is, this size is the pin, small pin, which and the tip of the pin. Okay, so this is the size of the zebrafish at the development is the pinhead size, very small, no, like a mustard seed. It's the size. Within that itself, you can see within 5 days of fertilization, total organ development. So you can have the live fish under the microscope, and a small petri dish can have that in a agarose embedded, and then the confocal has an immersion objective, which you can immerse through the water and keep it over the zebrafish, and then you can see everything in a screen. Okay, when transgenic label, the heart development, or eye development, or thymus development, everything, or kidney development, everything you can make a video of it. Okay, once it is a transgenic label, that's advantage. Now coming to a few techniques. Gene knock-in. Gene knock-in. How to knock-in means putting a gene inside an organism. How to do that? I'll explain my experiment, how I did that experiment at Yembel. So just show that if this is a protein molecule, it will have a gene, normal. Okay, so here is that. Now, you see, if you want to find out now where is this protein molecule expressed, what you can do is, this is a molecule called GFP, or green fluorescent protein. You can take for the gene for GFP, okay, and insert it before this protein molecule, the gene for this protein molecule. So what happens is, when this is expressed, this expressed, along with this protein molecule, a small GFP also will be expressed. So so once you put this fish in the microscope under a UV light or a particular laser wavelength, this GFP will get glow. Then you know that this is the site where this protein is expressed. So this is a gene knock-in. Okay, the GFP gene is originally from a jellyfish, and not from the zebrafish. They don't have. Now the techniques, vectors have come already. Lot of work is there where you can clone a gene for GFP along with a protein of interest and put it in the genome. And this is what I did at Yembel. I'll tell you. So transgenesis, or a gene knock-in experiment, which I did with Medica. This is a little bit more advantage model than zebrafish, because it's highly transparent. This is called the rice fish. Even we have we have collected some from some rice fields in Chennai district and maintained in Medica. It's highly transparent, and the genome size is half. So a lot of functional genomic studies can be done in Medica compared to zebrafish, and many laboratories also use it. So because in the lab at Yembel, Professor Whitgold lab, I did my Mercury project, and then because they are using zebrafish in this Medicus model, that was more advantage for me to use Medica. Now, how to make this Medica transgenic? Okay, one is you need to construction of vector. A vector, you know that. I had constructed a membrane GFP, nuclear RFP vector, then go for microinjection, production of transgenic fish, and 4D imaging. Finally, confocal imaging of the expression of this fluorescent proteins. So coming to fluorescent proteins, there are two types of fluorescent proteins I used. One is called the green fluorescent protein, red fluorescent protein. So the properties is that this can absorb wavelength at 488 nm, this green fluorescent protein, and re-emit at 514 nm within the visible light range also. We can see that. Okay, under the microscope, you can see the green color. Then the red fluorescent protein is a small variant of green fluorescent protein. It can absorb waves at 514 nm and re-emit at 633 nm. So wherever red fluorescent protein, that we can visualize. Here is the red fluorescent protein. So this is the properties of this protein. So what I did was I-Sec1 vector. This was prepared by me. This is more, though this is compared to CRISPRCas, this is a little bit old technology. But when I was working over there, so if you want to get more details, you can go to Graber and Whitbread paper, highly efficient zebrafish transgenic mediated by meganucleus I-Sec1 vectors. This is the Whitbread lab only, Professor Whitbread lab. I did my work. So I did first is I did a I-Sec1 vector like that with the gene for RFP, red fluorescent protein. So the gene and this vector has because I want to study myocytes, muscle development. I took the alpha actin. Actin, actin and myosin are expressed proteins in the muscle. Alpha actin is another form of actin molecule. Took the enhancer and promoter. The enhancer and promoter. And promoter is a short segment of polynucleotide, which is the coding, which is near the coding sequence of the alpha actin. Enhancer is something upstream, a small coding sequence which promotes the transcription, transcription of this more number of alpha actin mRNA. Okay, so this is also this enhancer is taken, promoter taken, fused together, and put it along with the gene for GFP. And same way, here is also another nuclear localization signal. A small portion of nuclear localization signal is also there. So whenever this is expressed, this will go and bind to the nuclear membrane, okay, the RFP. And here is another vector produced, alpha actin, again, same thing, but a GFP. This is a plasma membrane localization signal. So this will go and anchor in the plasma membrane of the muscle, and not in other cells. There are more than 200 types of cells in zebrafish, and only muscle cell expressed. So very specifically labeling, only the muscle cell is the objective of the study. So put this, so two vectors it took about 3 months, test the vectors and then putting cloning and this thing, and finally testing and finally vector study. Only after microinjection we'll know that whether it works or not. So the principle is, as I told you, this vector, if it is integrates, it produces this protein, okay, RFP protein, okay, and then anchors in the nucleus, okay. This vector will produce the GFP protein and anchor the plasma membrane of myocytes, muscle cells. So this is what happens. Red, then the nucleus will be red color only in the muscles. The plasma membrane will be green color only in the muscle. Okay, and okay. So this is how it is done. If you see that the zebrafish, we have to put the medica. If you put it before a day before, you separate the male and female. Okay. Professor Whitbread lab has 2,000, more than 2,000 tanks. You can imagine, 3 technicians take care of all the tanks, 2,000 tanks, and about 15 members in the lab. They use this 2,000 tanks for different type of experiment. So so whoever want to do the experiment, you will be given some racks, and then you have to go and separate about 5 or 10 males and females separately. In the morning, and then you put for the mating. Automatically they'll mate, and then the eggs will be in a clutched form. You will get and the females coming putting out the egg. Okay, fertilized egg. Okay, then you collect the egg. Each will be small size, mustard seed size. Bring it in the lab. You put that in ice because you have to prevent the fast replication. Okay, the blastoid disc like 2 substrate, 4 substrate, it will go very fast. Once you put it in ice, it will slow down. So because you need to inject the vectors. okay, under the microscope. In the blasto disc, you have to inject the vectors. So you don't want to have a 32 cell stage or 64 cell stage. You want the blasto disc slowly developing. So that's what you put the time in the small tube, petty like a the end of tube. You have the vectors ready. A very small quantity of vectors, few nanoliters. So now this picture I was on. Hirako was my friend, Japanese friend, who was also working in a. So he did some this thing, a picture shoot for me. So you have to arrange the eggs, small eggs in a small petri dish, okay, because and that arrange in a groove made of agarose, so that while injection they don't dislodge. Otherwise the water they'll be keep moving around. So injection. So make it ready. Make the tip ready, and the micro injection needle. You have to make the needle ready. Already with the needle puller, you have the needle. Now take a small aliquot of this thing and put it into this needle, very thin needle, very small needle, and it will be a very small opening. Okay, it will be there. Load that. It will be enough for 100 injections. Once you made that, then you have to mount this needle here, and the bottom you have the plate which has the small trying to develop embryos. Okay, where you can have that. So it's a robotically injected. With hand you cannot inject because of a very small pressure you have to give and inject into the blastodisc, developing blastodisc. So few injections we made, and then almost in 6 to 8 months, I think, we got many embryos, and finally you got the fish green. You know that if it's green, and the GFP is expressed in the muscle. Okay, you cannot see in the eye or brain the green color because it's only in the muscle. Alpha actin gene is expressed. So along with the alpha actin gene, this GFP will be also expressed, and also RF will be expressed because this vector has the promoter and enhancer only for alpha actin. Okay, which is expressed. So this is all we got. We did a gene knock-in experiment to make a transgenic Medicus. And now, this embryos can be going for confocal screen. So if you see the picture, this is one picture of the developing myocyte. This is not from the muscle. Okay, and if you see that, if you see the custard shape, if you divide into 20 portions, 20 portions, and then one portion is this area. Divide that into the pinhead size. Divide horizontally into one portion, 20 portion. One portion is this muscle developing. You know that why red color of the nucleus because that RFP is expressed in the nuclear membrane, and green color is because of the GFP. Now, confocal microscope, the advantage is that you can keep the live fish here, okay, in a water medium, and hold it for maybe 48 hours, 2 days, or even more than 48 hours. You can just image it, everything. And here, this can take XY, you know that, single shot, X direction, Y direction. Then you can go stack by stack, okay, one layer after one layer after one layer, okay. So that's called a 3D microscope, okay, 3D image, okay, you can. So this is exactly a 3D image. It's not a single image. Many images were taken, the same thing, like 1 meter, 1 meter. Then all put together, you get a 3D image, okay. Or you will get only, if the first shot only a little bit of red and green, you don't get the full picture. So this is a 3D image. Now, same thing you can go over time. Every 5 minutes if you want to image. For 2 days you can image. That's called the 4D microscope, okay, or 4-dimensional imaging. You can get a movie, how this muscle is formed and this thing. So this is the next part for muscle-related disease, and then molecules which can enhance muscle development. We don't have in the market molecule if you want to enhance only your hand or your this thing, some muscles. Okay, you don't have some medicines, or if there's some injury in your hand or muscular dystrophy or something, we don't have a very specific molecule. So we are planning to go further translational research using this model to find out the muscle-related disease, small molecules or targets in this in the myocyte, which can be used as therapeutic targets for further disease. This is my work. Okay. Now, coming to a few techniques. told about by gene knock-in. Then another technique is that in situ hybridization. So if you want to find out where we have 20,000 genes, no? Okay, where is this genes expressed? Where are individually where they are expressed? You do an in situ hybridization. Okay, this is also called as a antisense rRNA. Okay, experiment where you can have using vectors, you can have an antisense rRNA probe expressed or produced. Okay, so imagine if this is this identification of stem cells using in situ hybridization in zebrafish. Okay, so some stem cells express a particular gene called GATA1 gene. Okay, GATA1 gene. So the gene for this GATA1 is taken and put it in a vector. Okay, which is commercially available, and then it will produce a small mRNA, like mRNA, which will be antisense to the normally expressed GATA1 gene. Okay, so if A is there, this mRNA will the same place produce T. If G is there, this mRNA will produce G. Like that, this will be an antisense probe which can be produced, okay, in a small test tube, and then labeled with a decoy, a marker molecule, no? Which can be. So once zebrafish is taken, you can fix the zebrafish and then treat with this GATA1 antisense mRNA. So this GATA will go and bind to the sense mRNA, which is wherever it is expressed, whichever cell. So here, like then with the staining, you can find out, and this notochord region, this GATA1 gene is expressed because this antisense goes. So this is called in situ hybridization. So like this, you can do any number. Okay, for intersegmental FLRK1, different genes. No, for notochord, this is a transcription factor. Exactly wherever in this notochord is there, this SH antisense mRNA will go and bind and give it. So this is the in situ hybridization, which gives a whole idea of gene expression studies. Now, two aspects are there. One is gene knockdown and gene knockout. Okay, knockdown means for some time you can knock down a gene. Okay, so that the protein molecules are not expressed. Okay, and another thing is knockout, completely removing the function of a gene from zebrafish. So this what I'm going to tell is gene knockdown, or maybe for a few days, no? When the embryo is developing, knockdown. So you can now what is the significance? You can identify what are the phenotypic effect of this gene. Okay, so if you knock down a gene, if the no fin is coming out from the zebrafish, then you know that this gene is involved in the fin development and translate into limb development in vertebrates. Okay, so something like that. So if you knock down a gene, if eye is getting defective, okay, you know that this gene is very much important for the like development of eye, like the brain, heart, things like that. Okay, so how it's done? Here, like it's chemically, it's something called morphol. This is an antisense nucleotide, which is injectable. No? You can take any gene, okay, like an mRNA for a gene. Based on the sequence, you can develop a morpholin, chemically modified. Okay, same way, whenever you inject this thing, this goes and binds to the mRNA molecule, blocks the translation. Okay, this molecule. So it will be gene specific to particular mRNA. You have like 20,000 mRNA. Any mRNA you can make it, and then you can same way what I did micro injection. You can inject into the embryo at the blastoid this thing micro injection. This will go and bind to the normal mRNA molecule, a particular one type of molecule, and then block its expression. But there's what is a translation into protein. So for a few days, this will be knocked down. So they also use a dye to inject, so to see whether, along with the dye, you know that the injected has passed on to the embryos. Okay. So this is the morpholin of this gene knockdown experiment. Okay. Now this is the CRISPR-Cas, a very very versatile and highly advanced method of gene knockdown. Okay, completely you can remove the gene. So if like a coding sequence of a particular gene is done, like even the muscle protein or some transcription factors is there, you can now whole genome is sequenced for the zebrafish and Medica. It's already in the database. So by sitting in your computer, you can find out if your gene, if you want to study eye development or heart development, you can take down from the bioinformatics. You can this gene is in the pathway. We want to find out the phenotypic effect. Okay, where and all this protein molecules is not eating. So what are the things damage it is bringing? Then you can use a knockdown. Take this mRNA. Like then you should produce this all vector based. No? The specific kits are there where you can have a single guide RNA molecule. So here about 4 types of single guide RNA molecule for the same target. A different region is taken and then pulled. Then you get the sgRNA, 4 types of sgRNA along with the Cas9 protein injected into the yolk region of this fish. Okay. Then you need that, particularly if you for one MIF or any transcription factor for present in humans and also in zebrafish. If you use like this method, you can have a total knockdown of a transcription factor. Then you can see the phenotype in the embryo. Okay, you can visualize the video. So this is the very versatile technology. You can go through. I took this example from View et al. Development Cell, 2018, where like using this technique in zebrafish. They have done the mouse. They have a lot of studies. Zebrafish very recent, only 2 years back, very good studies are there where like 100% gene knockout models can be developed, and this has tremendous application biomedical research. 100 genes are in eye development. You can knock out and find out that. Then trying to rescue the knockout with small molecules, lot of entrepreneurship opportunities are there. I'll just come a little bit later. So now coming to the application, just excuse me. I'll try to explain. In biomedically, what are the different models used for screening in the drug discovery using the zebrafish? Okay, so now see that in the cancer, the type of cancer. Now this is a picture taken from Facebook. A tumor cell creating its own blood supply. So this is a tumor cell. So it will somehow get connected to the blood vessels. Okay. Then because blood flow is one thing which is very much needed for the tumor to grow. So it will just create its own vessel, connect to a blood like a vein or artery, and then pull in the blood, okay, and then keep on growing. So in a way, if you can inhibit this artery is getting formed, okay, artery is getting formed, then this is a potential method whereby you can destroy the cancer cell, because once the cancer cell is forming, lot of arteries will put. If you have some molecules blocking the arterial development, okay, then that is a potential anti-cancer molecule. Now using zebrafish, how this is done in a large scale. So this is a zebrafish embryo, developing embryo, 12 hours, 18 hours. If you see that this is an autocard, and now like a small vesicles called intersegmental vessels, that's a blood carrying vessels form like that, in the notochord. Okay, so and now this development process we can inhibit. If we screen for like 3,000, 4,000 molecules, some molecules, if we can inhibit this process of forming this intersegmental vehicle, these are potential anti-cancer molecule. Okay, a potential hit molecule which can be developed as lead and then go for preclinical study, etc. So this is done in zebrafish angiogenesis screen. Okay, now what they did, they developed a transgenic model of zebrafish, using same method as I told, transgenic model, FLI1. This is the marker for this the small vessel forming. It specifically expresses in the blood vessel. So these are the images taken in the zebrafish. And because everywhere the vessels are formed using computer generated image like segmentation. Okay, then they can segment this image, and finally they can segment the different organs and select only this ISV region wherever this blood vessels are forming. You can select. So particularly the program will quantify this ISV development in the transgenically labeled zebrafish. Now, using this thing, you can go for, because you can have that under the microtiter plates, thousands of embryos. The objective will scan each embryo. Okay, then quantify the ISV development, and then you can add drugs to the different screens. Okay, find out which drug can inhibit this ISV development. So that drug is a potential anti-cancer drug, because that can inhibit the angiogenesis, the process of angiogenesis. Okay, so this it cannot be done in a mouse model, because you cannot see the mouse under the developing the microscope. Now zebrafish means like a lot than you can do, and then this is an advantage of the transgenesis in the zebrafish. So particularly you can come out with the anti-angiogenesis molecule. Now, that is anti-angiogenesis. Now, here is another model where, like, tumors are developing. Tumor, particularly in thymus. How to find out a molecule which can prevent tumor cell in thymus, not T cell formation. Okay, now what they have done is they have labeled the embryonic zebrafish, the thymus development, T cells with a GFP. The same way method. Take a marker, put in a vector. Okay, that marker protein is expressed only in the thymus, along with label along with the GFP. So only in the GFP. Now the principle here is that when the zebrafish develops, all the organ develop. So the multiplication of the particular T cell can be compared to the multiplication of the cancer cell. Okay, because same way it is multiplied. T cell multiplies and then forms the organ. Okay, here also cancer cell already formed organ, then it keeps on multiplying. So any molecule which can inhibit this T cell multiplication can be a lead molecule for the anti-cancer molecule for thymus, acute lymphoblastic leukemia. Okay, so so this is the potential molecule. So now what they do is they have this thousands of zebrafish labeled and screen for molecules which can lessen this green color. Okay, so in some of this tray, if you have a lessened green color, you know that here the T cell is not fully developed. Okay, or less number of T cells are developed. So somewhere the process of T cell develop or multiplication, a molecule is going in some pathway. There are hundreds of pathways. So some pathway this is going and inhibiting. All other organ development is normal, but only this T cell is like having a abnormal, less than the green color. So in this way, this group, Ridges group, screened for 25,000 molecules, purified molecules, 25,000. They found out that one of this molecule, 1H indole 3 carbaldehyde 8 quinol hydrazone, which they call linyl dichar, LDH, is specifically able to ablate the immature T cells, or potentially it's a good drug candidate for T acute lymphoblastic leukemia, T acute lymphoma. Now, this is the hit molecule. At a target, that's a protein molecule. Hit molecule, in the industry, a novel hit molecule can get up to up to $10 million. Okay, or about INR, 20 crores to 75 crores is the value of a good hit molecule. Good hit molecule. See, hundreds of hit molecule, hit molecules are already available in our plants and animals and this thing. With the specific assays, you can develop or have an entrepreneurial objective to finding novel hit molecule. This is one example. Now, this is a more advanced technology where earlier we are telling this all marker genes. Okay, all marker genes are zebrafish genes. Okay, but they have orthologs, orthologs in humans. Okay, almost similar genes in humans. Now another group of scientists, what they did was they took the human gene itself. Okay, here is BRAF gene, and then put that into zebrafish. Okay, now screening will be very specific to humans because human gene is there and put into somewhere in that particular cancer molecule, inhibiting the cancer. It will be more relevant to humans also. So they zebrafish with the human cancer genes. What they did was they took this BRAF gene from a human and then made a transgenic BRAF along. Then finally, if you see that adult, only this BRAF could make a tumor. Okay, tumor. These are the controls. These are the positive controls. So this is used for screen. Now this will have a direct bearance on the humans because this is the human gene getting expressed. Okay, so whatever pathway some molecules can bind and lessen this tumor, that will be direct relation to controlling the human tumor with this using this BRAF gene or humanized zebrafish. Okay. Now, repurposing of drug, one group could find out, a lot of entrepreneurial opportunity because many drugs have cleared this FDA, toxicity and other preclinical tests, which are already available. Okay, with many firms. So, if you come out with a drug from against this already known drugs, then it's easy for other approvals. No? So, almost 50% of the 15 years, 17 years, 7 years, you can just let off because this is already preclinical studies have been over. But a new application. Okay, so here one group they have found that this BRAF inhibitor, they found that the target is dihydrogenase in an enzyme. Here is which can bind and lessen this tumor. Okay, that's a BRAF inhibitor. So this is under clinical trials. No? Leflunomide is under clinical trials. So this using zebrafish they found out. A drug can be used for one of the human cancer. So this is one example. Now a company called C-Clinics. Okay, C-Clinics in Switzerland. Spain, I think. Okay, they are doing a lot of work, and this is a lot of opportunities in India also. No? Personalized cancer treatment. What they did is, so if you identify a cancer patient, there are maybe 100 drugs there to cure, but you cannot keep on trying each one drug. Which drug is best effective for this patient? You cannot try because time is very short. Okay, so this company what it does it takes the biopsy of the cancer cell and inject into the cell into the zebrafish embryo. Okay, so if it is like a sarcoma, like or some leukemia, no? it will go into the system, and some of the blood cells will get this human cell and keep multiplying that. Once it is labeled by GFP, you can see it. The human cells multiplying inside the zebrafish. Now, like if you are testing 100 drugs, you can test and see which drug has the capacity to inhibit this human carcinoma cell which is developing inside the zebrafish. So 100 drugs are there, then 2-3 drugs will be very, very effective. Then in bringing the red color down or destroying the human cells within the zebrafish. That can be directly applied to this patient because it is his own cells which is taken and screened. So this personalized cancer treatment, I think in the few years, has a lot of opportunities. You can have startups in India, like on this personalized cancer treatments. Even I think every state can survive a good startup in this personalized cancer treatment because cancer is one thing which a lot of money people are spending for cure. So enhanced cure people will be looking for, and this is a good opportunity. And even this company has a drug in phase 1 and phase 2 clinical trials using zebra for epilepsy. You see that now they have modeled zebrafish for epilepsy screening, and some small molecules have been screened, and not given the details, but phase 1, phase 2 trials can be used from zebrafish screen. Then coming out to the drugs for kidney disease. No, okay. So like same because 78% the humans the orthodox in zebrafish, they could find that like on in the kidney regeneration, LHX1, okay, or Hobiobub's transcription factor is very much needed, okay, and if you want to damage kidney, if you want to regenerate it, automatically the marker is LHX1 gene. Okay, automatically this is getting enhanced expression. You know that kidney is getting regenerated. Okay, so zebrafish, they have done a development high content assay for kidney progenitor cell expansion in transgenic zebrafish, which is this LHX1 is the marker. I think they have the picture. Okay, here it is. No, in the zebrafish, the marker. So if some molecules, one can enhance this expression of the marker, they are the best suited candidate for this kidney regeneration. And I just went to see that, like in the kidney disease, these are the different diseases, Van Meltingen syndrome, like renal hypoplasia, renal bilateral, something like that. So many types of kidney. So in a way, like to find out the effective drug for kidney itself, kidney disease alone, we need 20 types of zebrafish. If I know, like maybe 1 or 2 types is there. Even startup only on this having transgenic zebrafish for kidney disease will have many clients. Can come out with new molecules, or big companies will have a partnership to give you their molecules, their library for screen, and come out with some lead molecules. They will pay in different ways. 5 ways of payment are there. The last slide I am going to explain that. Hard to repair. Okay, same way. Transgenically labeled zebrafish, and then test with the different drugs, and they found out that they out of the third 3,000 compound screening, visnagin and diphenylurea are cardioprotective compounds. Okay, so the target is the protein called malate dehydrogenase. Okay, which represent a novel and druggable target for cardiomyopathy. So they found out a new target also for human disease in zebrafish. Okay, that's a very, very phenomenal work on this heart repair. Then screening for cardioprotectants, using zebrafish, because everything you can, even in one of our study, we could find out. Dr. Rajesh Khanna will do my PhD. He's a group leader in Satyababa University, nanotechnology lab, doing a lot of work on zebrafish, because you people are near, you can always visit Dr. Rajesh Khanna lab in Satyababa University. A lot of he has transgenic lines also. A lot of work is being done in his lab. He did a work in our lab, Tyson, using cardio protectant assay, where you can say have the zebrafish, and with a small microscope, lot of screening he could do, and we isolated a molecule, not highly purified, a molecule from one of the herb. Okay, cardioprotectant assay. Now diabetic drug screening. You see the pancreas of the zebrafish. Okay, though it's small. Okay, same beta cells, alpha cells. Okay, delta cells, all expressed in the pancreas of the zebra. So any here they label separately, like beta cells green and the alpha cells yellow color. Okay, so that they could see the different like multiplication of cells. Also in the pancreas, like the degeneration of the beta cells, which produce insulin. So if we can find out some molecules which can enhance the multiplication of this beta cells, that's a potent anti-diabetic molecule. So this group, Joe et al., could screen 83 molecules and identify 20 lead compounds. Retinoic acid, glucocorticoids increase the beta cells regions. Using simple transgenic zebra. Because if it's not transgenic, this you cannot see. Okay, so that you can have the zebrafish, but everything will be transparent. And it's because it's only transgenic to only beta cells and only delta cells. You can see that, visualize under the microscope. That's the advantage. And also the animal is live. Same way, trying to explain. This is a picture, no? Green color giving beta cells in yellow color and delta cells in the red color. Okay, so different. You can get lot of data and coming out with meaningful results. Okay. Yeah. Now this is also another very good example where here we were telling about all hit molecules. Target discovery. Target, as if you see, recollect the earlier picture, that's a protein molecules. Now, in one of the sarcoma, okay, one of the most common cancer in children and adolescents is rhabdosarcoma, cancer of soft tissue such as muscles, tendons, or cartilage. Okay, here they found out in humans that fusion of DNA of two different chromosomes caused the most aggressive form of rhabdosarcoma. The first DNA product produces an abnormal protein called PAX3 FOXO1. This is a transcription factor. PAX3. So once this genes get fused, okay, or mRNA, a fused mRNA comes, and that produces this protein, which brings in cancer. Now, this group, Kendall et al., could isolate this protein, okay, then put it with the GFP, and then inject, transpose is immediately injected. Then they could find out wherever GFP is expressed and the PAX3 FOXO1 is expressed. They could isolate the tumor, okay, and make studies. And then they found out that HES3 is the HES3. It's a protein molecule which is the target for this cancer to develop. No? Human HES3, the human ortholog of HER3. Okay, the same protein which is present in the humans is called HER3. Here they used differential gene expression. They took two sets, no? Made a PAX3 transgenic fish. Okay, then isolated only these cells. Okay, isolated only these cells. Okay, then another this thing with FOXO1. Okay, isolate only this. Then go for FAX sorting. Okay, FAX means fluorescent activated cell sorter. Only cells. What are the mRNA coming? Only this type of cells, red type of cells. What are the mRNA coming? Then analyze all the because if it's a cancer, then one type of protein will be highly expressed. Cancer. Then they could find that okay, HER3 is 21 times more expressed. HER3. So now you know that this is one of the target. Okay, because this is highly expressed. So in a way, using zebrafish for one of the human cancer, they should find a target. So in the industry, target could fetch up to 10 to 30 million US dollar, because 75 to 225 crores is the value of a good target, because so many companies will license from you, or you can sell your target if you have a startup. No? The 3-4 years you work and come out of the target, you can sell your target. Though a little bit theoretical, it's possible. No? Once you have the knowledge. No? These are knowledge-based industry. So a PhD student or young group, no? You have to think on that and have a good knowledge. You have a strategy. Collaborate with people. Okay, write to people. This is my idea. This target. Then slowly you'll see that 2-3 years you will have some data, some patents filing. Then go for a startup. Okay, automatically you 5 people are there. 5 people is enough. No, you can find maybe a hit molecule and target molecule through startups. So this is the zebrafish-based target. This is very new work, and I know like maybe 200 or 300 human targets are yet to find out. So a lot of scopes are there to find out novel targets using zebrafish. High throughput screening is a robotic screening where zebrafish can be used. Why zebrafish means like a high fecundity. Lot many, if you want to have 20,000 embryos in a single day, you can have rapid screening, because in 2-3 days all organs develop, all neuro behavior and disease modeling. Genetically you can trace it, no? What are type of genes and this thing. Okay, so then robotic screening, you can find out hits or targets using zebrafish. Now go through this article, no? By Bugle. It always gives a whole detail of high throughput screening. This is one paper which is which fascinated me. You know, this is a pin, small pin. This is a pinhead. Okay, you can imagine the size. You can have it in a like a agarose embedded, keep the animal live. No, this animal can be kept live for 2 days. The pinhead, you have using a light sheet microscope. This is a new novel microscope. You can image the total brain activity. Okay, same way you can image also any organ activity 3-dimensionally. Okay, it's not a confocal, it's a different light sheet microscope. So I used high-speed light sheet microscope to image activity 80% of the neurons. No? About 100,000 neurons of a fish, the real-time activity. So even lot many studies can be done, or drug screening can be done, which affect the neural behavior. Or even Rajesh Khannan team, recently we had one of the PhD students expressing the amyloid plaque proteins in the zebrafish. So lot many studies can be done using or finding a cure for Alzheimer's, etc. using zebrafish and also light sheet microscope. Now, coming to this part, I have finished with all the screening methods, no? Using zebrafish cancer to all the diabetic heart disease. Now, coming to FDA, zebrafish assays have been accepted by FDA for toxicity and safety assessment for investigating new drug approval. Okay, so 2014. So this assay is used or accepted. No? European Union, they also accept this zebrafish because they see that up to 5 days, these embryos are classified as in vitro models. Okay, so no issue with the using of animals up to 5 days. Then up to 5 days, okay, you can get the permission. Is that so? You use also recognizing zebrafish as a good model. Okay, so now we'll go to the third part of my talk: entrepreneurial prospects. Okay. See that where are the entrepreneurial prospects? Identifying novel human disease targets, identifying human therapeutic drugs, novel peptides in organ growth, organ repair, prospects of mass production, biosimilars in transgenic. How much is that? Is like a global market by 2025 for biotechnology or transgenics, everything related is the market. No, market is $729 billion. 1 billion is like almost 7.5 or 7,500 crores. 1 billion. Okay, so this is the market for biotechnology, drugs or screening or personalized care or like the so many things. Pharmacogenomics, so many things are there. It is the market. No? You can see that it's global market, not Indian market. Okay, now if you see this, if you divide this, it has drug discovery, has clinical discovery, preclinical, laboratory service, many things are. By 2025, okay, this is almost 90,000 million, 90 billion US dollar market. Particularly, startups can emerge in discovery area and preclinical area. How much is the 15 billion US dollars? That is 1.2 lakh crores the market. So if 10 companies can come, already there are more than 1,100 companies will be there. Preclinical already there will be 100 companies. Okay, but there are space for new startups to come and earn like 5 crores or 10 crores per year for the revenue. This is the space, not discovery base. You can aim like 1 million, maybe 7.5, 5 crores, no 7.5. So this is the space where you can plan for startups. Okay, using zebrafish as model. Okay, so they will be a contract research organization for major pharma. Where are the opportunities? Like specifically, even a company can produce zebrafish embryos and sell to different labs in India. Okay, just embryos, not transgenic. Then you can have companies which can produce antibodies for zebrafish. If you have 20 or 30 antibodies produced in zebrafish, okay, and against zebrafish, but that's a sellable product globally because there are very, very less number of antibodies. Then a company can produce zebrafish knockouts. Only few knockouts are there. If a knockouts are already drug discovery companies will be maybe ordering for 10,000 knockouts or 20,000 knockouts. Our research labs around the world will be ordering for a particular knockout. Just send us this much number of knockouts of particular model. Many number. Then zebrafish preclinical, whatever I told the examples. If you standardize examples, companies will be giving you molecules to test preclinically. Okay. Then hit discovery you see. Hit means small molecule discovery you see zebrafish. Target discovery. Okay. I am very sure that in this area, for new startups to come, at least 100 startups can come, zebrafish based startups in India by 2024. I'm very, very positive on that. Okay. I'm a die-hard optimistic thinker. Okay. So with the knowledge based and people are very rightly doing, we are trying to develop a few companies. My one student, one Indian is there, is in like Ash Batak and many students from our lab and this thing. We are planning some from startups on this there, but maybe we can do only 2%. No? Another 98% people listening me, you should think 98%. Can we bring a startup? No? Starting with the minimal investment or trying to talk to angel funder or startups funders. No? Maybe because angel funders try to take risk. They can put easily 20-30 lakhs under a team. For a single person, they do 4 or 5 people who finish the PhD and good experience, maybe postdoc, 25 to 30 lakhs for a salary, trying to come out with some technology on this area. Angel investors will surely put it. If we approach 10 people, one of them will put like 25 to 50 lakhs. Our Government of India funding is there, DBT funding is there. If 5 of you there, surely you'll get 50 lakhs for ignition grant and things like that for a specific knockout preparation of the zebrafish or something like that. Will be for sure. And if drug discovery startups are there, as I told you, novel target. No? Okay, so the pictures reversed. It can give 3 to 10 million. And your target can give 20, 30 million. Okay. Who are the clients? The major global pharma, no? More than 20 pharma are there. You can be a contract research organization for this CROs for this pharma. Whatever they give us upfront payment, licensing payment, R&D payments, milestone payments, royalties. So if you have developed some five zebrafish model, you took a grant of 50 lakhs and I'm sure that some company will pay you the total R&D cost. Okay, and license your zebrafish for their study. Okay, it should be new, no? New type of models, which is not which these companies don't have. Okay, then they'll give milestone. Once you have a small small molecules, like molecule from the plant that is very effectively controlling some cardiomyopathy. Some of these companies will give milestone and they license your molecule or buy your molecule, or they put money in your equity. If you have like 100,000 shares and your company is valued at 1 crore, then they will put equity and value a company up to 5 crore. Okay, for a startup company. Many things can be done. There are many opportunities, and people are looking forward to have a very good technology which can have a global reach. I am sure there are 700 funders in India, angel funders. Somewhere I just saw that thing. 700 funding people who are ready to fund for good like a project. Not exactly not research and it should be product oriented. Okay, and at the end of 3 years, you should have a molecule to sell. You should have a target to sell. Not piling out with paper. Maybe only piling with patents, no? 10 or 20 patents, and then the patents will only fetch money. Okay, not the paper. Okay, so be clear here. No? Okay, only product. Okay, first is patents, then that will have the product tied to it. So this only will fetch money from the angel investors. Once you have the angel investors ready, then you can have the venture funders come in. Series A funding is there, Series B funding is there, C funding is there. Like that, your company startup can grow from scratch, from idea to a good company. There are it's not like a bed of roses, but there are a lot of challenges. But with knowledge, you can break the challenge. Okay, that's important. With knowledge, you can. And trying to find out the right people, you can break the challenges and come out very successfully. Okay. So there are different other entrepreneurial prospects. Identification novel peptides. So these are the different heart, lungs, liver, bladder. etc. Except for a few molecules like erythropoietin, insulin. And we don't know like a molecule like which can maybe I'm telling the biological molecule. Okay, not small molecules from extraneous source like bacteria or plant. Our own molecule, small molecules like peptides, which can increase the restore the function of the pancreas, restore the complete function of the lungs. These are not found. Okay, our own molecules. No? In the few coming years, it will the small novel peptides are growth factors. Okay, which are very specific to liver, bladder, kidney. This need to be found out. And zebrafish is a good model. Where you can find out these small novel peptides, which are very specific to organs. So novel organ growth factor. That's what I call. Okay, this is one area of our interest. Like this thing, like finding out novel organ, bone or bone marrow, things like that. So that will be directly applicable to human. You can find the ortholog of the human, then it will be directly applicable to human. Okay, here also, and the humans 3D printing of organ. Okay, because in the next medicine is that we all need a heart. Okay, at 70 years or 80 years, heart is one thing. So if you have already from our own stem cell, if you have a heart ready made and kept, okay, then it's easy. Say you need a liver, then you can prolong your life for 20 years with your own heart, your own liver, or your own spleen, or your own bone marrow. Okay, so this 3D printing of organs is very, very, very much needed. And from the BRICS Biomed Consortium, this is one of our focus of the 3D printing of organ, because this has a global opportunity. So some of you start thinking on this aspect. 3D printing. Already people are different labs are making. Nothing has come to market. So this is a yearly area where India or Indian scientists can focus and then come out with good strategies of finding a good heart and good spleen, etc. Then one other thing is that mass production. We use like cell culture based. Even the zebrafish can be used mass production, biologicals, biosimilars, so that if insulin you produce human insulin, then the cost of insulin will come very, very low. Okay, or 10 times lower. It will be beneficial to the population, and a lot many startups also can come in this area. Now, to summarize, as we saw that, what are the different strategies in the drug discovery? How gene knock-in called transgenic fish can be created in my experiment, and then T cell, using T cell, how you can find out novel drug for leukemia. Okay, humanized zebrafish embryos. So how exactly very pertinent data, pertinent molecules can be pulled out using humanized this thing, and how using zebrafish, novel target is identified. High throughput screening using zebrafish embryos. Then what is the startup space opportunity for startups, and then how startups can come, and then who are the clients, and what are the areas they can earn the revenue. Just we saw about that. And now on tips, like always say that 3 tips per se. Read something no one else is reading, think something no one else is thinking, do something no one else is doing. This is important. Okay, for your success, for your newer ideas or startups or this thing. This is very important, because I always put that all this on. Now, survival of the fittest, we have heard that, but almost everyone is fit. It's the survival of the fastest. Whoever is going fast will survive. Okay. So now, like I am in LinkedIn. Okay, if you see, put SG Prakash Vincent, type in LinkedIn, and just give a connect. Okay, I'll be happy to connect with you who are interested, and we can have a discussion. And best of luck, and thank you all for Prakash Vincent. Thank you very much. Okay, look forward for the questions. Thank you. Hello.
Sir, thank you very much, sir. Sir, we have few questions. I'll pop up those questions, sir.
Yes.
So, which drugs are already prepared by transgenic fishes and for which diseases? Sangram
Yeah.
Kesari Panda wants to know, sir.
Yeah, this already I have given the few examples there. If you slide, let me see. Okay, this is a molecule called diflunomide. Okay, this is against the cancer molecule. This drug is repurposed and it's in clinical trials. Yeah, one example. You can
Okay.
share this slide. Okay,
Okay. Okay, sir.
so that you get. There are a few examples like Visnagil for heart repair. This is the drug which is developed using zebrafish.
Okay. Thank you, sir. The
Yeah.
same person wants to know, sir, in India, which are the institutions do research work towards drug discovery by using transgenic fishes? Which is the most common transgenic fish used for this purpose?
Yeah, I think not much. Not much. This, maybe if you can mail me, I can find out and tell. Because transgenic using zebrafish, there are a few labs I think using, but transgenic I'm not sure. Maybe Rajesh Kannan lab is doing a small part. Confocal microscope is one of the constraint, but not much. But if you can mail me, I can find out and tell. We are also planning for a few years. Maybe another 1 or 2 years we'll be able to start it.
Okay, sir. So, sir, generally transgenic fishes are done for eating purpose or medicinal purpose?
What is the question? Repeat that. For
Generally, transgenic fishes are done or created for food eating purpose or medicinal purpose.
Only for medical purpose, not for eating purpose.
Okay. Okay.
Only for medical. Okay.
Okay, sir. Pranay Chakrabarti has asked, are these fishes ecologically sustainable? And are these harmful to any particular species?
Uh, uh, because this is not left out in the wild. This is all experiments carried out in the laboratory. Okay. And then the protocols tells for the total destruction if you're not going to use it. So there's nothing compared to the ecology. So sustainability also comes into the question because it's all done within the laboratory, controlled laboratory conditions.
Okay, thank you, sir.
Yeah.
Sangram Keshri Pandas asked, sir, can algae be used directly for the treatment of disease found in these fishes?
Algae directly, I don't think. Okay. Well, algae has some like immune boosters and things like that. Okay, when you directly spirulina and things. But directly for one particular disease, I don't think.
Okay, thank you, sir. More than questions, everyone have thanked you, sir. They have, for the nice and informative session. Someone said for the first time they are hearing about all these facts, what you have shown.
Okay. How many participants were there total?
Sir, so far 680 people have attended, sir.
Attended. Okay. Good.
Yes, sir. So, sir, one student, how to differentiate a normal fish from transgenic? What is the selectable markers used for drug production in transgenic fish?
Yeah. This is this differentiator is what you inject the transgenic vector. If you see the particular color in the under the UV light, okay, under the microscope or under, then you know that this is the fish has taken up the transgenic character. Okay. Then the marker is like many places and like X1 and many places I have shown the markers for particular this thing. You can go through the slides and find out more than 10 markers.
Okay, sir. Sir, Dr. Anshuman Das has asked, by using these techniques, can we modify the size of appendages of animals, particularly for lower grade animals like invertebrates?
Yeah. That you can do. You can do. You can knock down a particular this thing, particularly if protein, they're going to transcription factor in the muscle, the animal will be bigger. Okay. Can be done.
Okay. S.K. Das has asked, are there any risk by eating these fishes? But you have told very clearly these are all used for medical purposes only.
Not for protein. Okay.
Okay,
But I
thank
don't
you.
think because whatever protein, the thing, it is overexpression of the protein. Like if 5 kgs of myosin is made, 8 kgs of myosin is made. The same protein is produced large, even if you are going for transgenic animals, it's like the protein is expressed in large quantity, same protein. So nothing wrong in that. There we don't use markers and things like that. Okay, because I don't think any the animal it has common marker, but it will come. Okay, it will come. And but as far as I know, there's no safety concern.
Thank you very much, sir. I think we have come to the end of the session. Sir has answered all the questions. Now I request our Dean Research and the convener of this webinar, Dr. J. Jayanti ma'am, to give her concluding remarks. Ma'am, please.
Thank you, Professor Prakash Vincent, sir, for the inspiring talk on the role of transgenic fishes in drug discovery and the scope for entrepreneurship. With the current situation of COVID, the importance on the role of biotechnology in diagnosis and health sector has gained momentum. Your talk on the importance of drug discovery and how this research could lead to employability and contribution to the society and to the nation was of vital importance. I feel a lot of students and upcoming researchers will be benefited by it. Thank you very much, sir.
Thank you very much, ma'am, for your words. Now, as a formal gesture, I would like to deliver the vote of thanks. I feel very happy to propose the vote of thanks for the national webinar on the role of transgenic fishes in drug discovery and the scope of entrepreneurship, organized by the Guru Nanak Center for Research, Guru Nanak College Autonomous, Velachery, Chennai, held today, 1st October 2020. I wish to extend my grateful thanks to our resource person of today's webinar, Dr. S.G. Prakash, Professor and Head, Center for Marine Science and Technology, Manomaniyam Sundaranaar University, for having accepted our invitation and sparing his valuable time and enormous knowledge and experience with transgenic fishes for drug discovery with us. Thank you so much, sir. I wish to sincerely thank our management, especially our most revered General Secretary and Correspondent, Mr. Manjit Singh Nair, sir, our respected Principal, Dr. M.G. Raghunathan, sir, for their constant support, guidance for planning and execution of this national webinar. I thank Dr. J. Jayanti, Dean Research, the convener of this webinar, for organizing such an useful and informative webinar. I'm thankful to Dr. L.R.S. Kalanidhi, Vice Principal, Students Affairs, Dr. Rajshri, the Controller of Examinations, the Deans of various schools, heads, faculty of various departments of Guru Nanak College for your participation and contribution. I thank all the participants who showed keen interest and very active participation in today's webinar and made this a grand success. Once again, thanks one and all. Dear participants, the feedback link for the e-certificates have been posted in the chat box. Kindly submit your feedback. Thank you so much.
Okay. Bye-bye.
So 1 minute, sir. I'll end the broadcast, sir.
Okay.